Thank you so much. My name is Doroteya Staykova, and I'm part of a small UK-based startup. And today I would present a pilot study based on the CAMDA challenge on gut microbiome health index. And it tried to address two main questions, namely how to integrate diverse data modalities pertinent to gut microbiome, and what is the structure, how it look like, this integrated microbiome from data perspective. But before going to details, I would like to thank the organizers of the CAMDA challenge for providing the datasets and the framework for making this study possible. So firstly, you already seen the data that have been shared, but I just want to recap again. So we have a dataset of more than 4,000 samples originating from 30 studies. So it's a functional and taxonomics data which provide a very large scale view of the gut microbiome. So it's very rich in information data. And as schematically shown in the middle here, we can present this data as two-dimensional feature planes which share their sample axis. And the metadata or the categories of healthy and disease is indicated with orange and gray color respectively. So when looking at this data structure, I could not help but wonder what healthy means in the context of microbiome. And I think it is important to answer these questions before comparing disease microbiome with healthy microbiome state. So to answer this question, I employed topological data analysis, shortly abbreviated TDA, which is its most popular algorithm, mapper, which transforms data or models the data as graph, as shown on the left here. So the first assumption that is made is that the data are sampled from a space. So on the left here, we see a data cloud in a two-dimensional space, which is shown as a scatter plot. So each dot represents a sample on the left. And then by taking the distance of each point to the most left located point, we can map this space into one-dimensional space, achieve one-dimensional mapping. And mathematically, this process is called applying topological lens or filter function, F. And by binning and clustering across the values of this function F, we can obtain, as shown on the right here, a graph visualization of the topological structure. I like to highlight the word topological structure of the original data cloud. So it's not really statistics here or geometrical representation, but topological. So then the mapper is actually, one can think of it, is like visualizing the shape of data through a topological lens, very much like a camera with optical lenses visualizes the scenes. So depending on the choice of topological lenses, different aspect of the data will be revealed. So going back to the CAMDA data, so if we imagine that, for example, taxonomics data are taken for a very large high-dimensional manifold with more than 1,000 dimensions, whereby each dimension corresponds to a single taxa feature, then with the orange dots are shown the samples that are living in this manifold. So then I used a diffusion-based embedding called PATE, with the full naming written on the top, which PATE is very good in capturing nonlinear structure in the data whilst preserving the local and global relationship within the data. And so this is the lens I use to project this very high dimensional data into the two-dimensional map, as you see on the right here. And immediately you see some branching structure on the healthy microbiome. And these branches indicate taxa-based subgroups within the healthy microbiome. and with the largest one indicative of a low Shannon entropy and mid to high health index. So then we can imagine that the microbial data, I need to highlight, this is microbial data only from the healthy gut microbiome. So it's a fraction of the data. And then I have, let's imagine they're embedded in a shared topological space with axes across both taxonomic and functional features. Then I have used two-party lenses and the mapper algorithm to construct a topological view of the healthy microbiome landscape, as it is shown in the middle here, whereby each node represents a cluster of samples, and they are clustered across both taxonomic and functional modalities. And the edges reflect the connectivity between, or sharing of samples between the clusters. So immediately you can see that two flares kind of stick out in this model, and with the blue, I don't know if you're able to see, but blue and yellow colored groups of the nodes, which when further investigating what makes these nodes or group of samples different, I use Kolmogorov-Smirnov statistical non-parametric statistical test that was mentioned in the previous talk. And immediately you see that these two groups exhibit significantly different microbial diversity and significantly different other healthy indexes that were provided in the CAMDA data as metadata. So further, when looking at the functional and taxonomics layers, these two groups also exhibited significant differences, whereby the blue group, for example, showed enrichment in P. copri, as shown on the top left there, and it's also well visible if you color the nodes with the enrichment of this species. In the model, it's well visible, this group of samples. The yellow group of samples, indicated samples, show more diverse composition, microbial composition with including presence of E. coli. And interestingly, this is something that is difficult to do with statistics, but in topology, if you color the nodes with the enrichment of E. coli, you can see actually a gradient across the flare with in the topological model, which is an additional layer of information. And of course, also on the functional level, there were significant differences as indicated with the violin plot on the right. So to conclude, we have, even in the healthy microbiome, diverse subgroups emerged, distinctly different subgroups emerged. And this challenges the notion of a single homogeneous healthy state. And also it's this finding suggests that a more data-driven healthy index that can account for complexity would be more appropriate. And please reach out, and I would like to thank very much for your attention.
Thank you very much for a very fascinating talk. Have we got any questions?
Hi, my question is regarding the lens function that you used. Well, why did you use that lens function? And I was also wondering if you made some other experiments with other projections.
I selected partly due to my experience with working with other data coming from clinical cohorts. I had done PCA projection, and you could see, as probably you have done already, it's kind of a cloud. It doesn't give you much information. So I wanted to, as I indicated, depending on the projection, you can unravel or untangle different layers of information. So PATE is non-linear, so you can use other non-linear methods, but yeah, for this particular pilot study, I selected this particular one to see if there are branches, because PATE is known for finding trajectories in the data. If they exist, if they don't exist, then of course there will be no such branches. So it's random walk based method, so it's very convenient when to test, to see another layers of information in your data. So this is the reason. But of course, other lenses can be used by all means.
Okay. Sure, thank you. It's very interesting.
Yeah, welcome.
Have we got any more questions?
Thank you very much for your presentation. I think I have a question along the same lines. Can you maybe tell us a little bit more about your understanding and feeling for this transformation, and to what degree it preserves local groups or local similarities? Because it is well known that, for instance, TSNE or UMAP do not preserve distances in a meaningful way. So then if you see heterogeneity in the dimensionality reduced space, that could simply reflect the fact that things that are close in the reduced dimensional space are not necessarily close in the real space, which again, of course, raises the question, what's close? What type of metric would you be looking at? So these are not necessarily questions for there's a single answer, but I would be grateful if you could maybe tell a little bit more about your thought process, because you've played with the data for quite a bit, I guess. So maybe you can tell us what metrics you've been looking at and what type of projections you were looking at, and what your insights were with respect to what type of relationships these projections either preserve or don't preserve.
Okay, I could talk a lot. It could take quite
Thank
a long
you.
time, but let's just wrap it shortly. I would encourage you to read the paper which published this method. This is not my method. This is published. And it is manifold-based method, and manifold are topological spaces. So I work in algebraic topology, so naturally I'm drawn to topology-based projection methods. UMAP is also topology-based projection, but there is a randomness in the UMAP. So yeah, I mean, you have very different directions to go. You can go lenses UMAP, you can go PCA, and in my experience, depending on the data at hand, different lenses will provide different information relevant to your question. So because so in this case, my question was, does the healthy microbiome has a structure? Does it have a shape? So this brought me to the lens which provided the most structural information, so to speak, going from manifold to projection in a robust, mathematically very robust way. So this was the reasoning behind. But yeah, I mean, I cannot explain the whole method because it's a whole paper and it's really not my paper. So I think other people should be explaining the methodology of PATE. But in my experience, I find it useful to see trajectories. So that's how this is also designed. If there is, especially for longitudinal data, if patients are going on different trajectories, this is very helpful to look at. I imagine also in microbial studies would help to see how the microbiome develops with time, trajectory-wise. So I think I encourage to use this methodology for data projections.
Can this be extended to use label information, like healthy, unhealthy, so that informs the projection in some way?
No, this is like UMAP. This is just a projection on the manifold using random walking. Random walk. Also used, yeah.
Thank you.
Welcome. I don't know if you have other questions that I miss, but yeah.
Each dimension is a variable? No, it's a combination. I don't know. I don't know the mathematics behind it, but each dimension is one variable.
So you mean the manifold? So here, the taxonomic manifold, I imagine it as each taxa feature provide a dimension. So you have more than 1,000 taxonomic features. So you have more than 1,000 dimensional space in which the samples live. And then, it is, so the idea is to look for projection that you can keep locally similarity from the manifold, but also keep the relationship between things that are close or far in the manifold. They should also be close and far on the projection. That's the,
There is no
sure.
directly translate down. No? If you see this big data, then not say this is because of species 1, 2, 3.
You need to do some statistics to define what makes them, but you can, this is entirely driven by taxonomic data. So the labels or the colors, they are afterwards, but they're not incorporated in the projection. It's the same with the topological model. It is driven entirely by the mathematics, and then you overlay on top of it the metadata to see if there is some interesting, so it's an exploratory tool to guide you which areas to focus on. Eva?
You haven't presented it, but do you have a think on whether the unhealthy samples have a similar?
I haven't done that. It's, yeah. Yeah. If there are opportunity, I would love to do that. But I think if for the disease, I think there will be somebody on board, clinician, who understand the disease before attempting even to dive into it. So, yeah.
I think there's one more question.
Would it help you to reduce dimensionality by, say, doing the feature selection for given health and disease states, to decide which features are important, and then reduce dimensionality and then do the topology? Would you have the same picture?
Would I? That's well, I haven't tried this. So in my experience, topology is quite robust to perturbances. So if those feature reduction do not massively perturb data, so if they are perturbing, I'm going beyond here. I'm brainstorming here. I don't have experience with this type of analysis to see if feature trimming or engineering would influence the topological model. I think this is a different line of investigation that I haven't done. But in my experience, using different lenses, so not feature engineering, but different projections, if the shape preserves, then this is a signal that is preserved no matter how you modify the data or trim the data or so pre-process the data, so to speak. Because for me, feature engineering or feature trimming is pre-processing, and I try to stay away from trimming the data because sometimes you enter the risk of losing important information. So, yeah.
You mentioned Privatella copy as driver of one of the clusters, and this triggered more general thoughts about how to handle all those individual differences. Because Privatella is a strong driver of known enterotype. So we're talking about enterotypes, individual clusters of individual structures of microbiomes, and I know that dealing with Privatella is typically a nightmare because you cannot easily distinguish between effects along health disease axis and difference between just in the individual structures of healthy people. And Privatella is a very strong driver. Unlike the majority of species, it typically varies from 0 level to like half of all the species abundance. And it's typically 0 or something.
Okay.
Not gradual change at all. Like that. So this is a strong signal, extremely strong signal between individuals. And while Privitella is often associated with like better health, vegetarian diet, etc., but it's still this signal by itself is much stronger than health disease signal. So we need somehow normalize or condition search for the health.
Yes, that's why I wanted to provide this contribution, because there is no disease. So this is really, to me, to establish the baseline for the community to not consider healthy as singular, to think about that maybe there is something more into the healthy, or where the healthy ends and where the disease. So this is the idea behind providing these results.
Okay, thank you very much. I think if there are more questions, then we can always just ask the speaker. Let's give a round of applause.